Assalamualaikum and good afternoon, everyone. Thank you very much for participating on our digital session today. So today we have Mr. Amir Muhaimin Akmal Shukri. He will be presenting on the construction of SARS-CoV-2 Delta variant virus-like particles and its application in development of enzyme-linked Aptasorbent assay ELASA. So without further ado, I would like to welcome Mr. Amir Muhaimin to start his presentation.
Thank you. So I hope that everyone joining us online can hear me. Yep. Yes. No. I think so. Okay. So Assalamualaikum and a very good afternoon to everyone, to doctor, to all the staff, and also fellow postgraduate students. My name is Amir Muhaimin. I'm a PhD student. So today I will be sharing a bit, not a bit lah, mostly about my study entitled Construction of SARS-CoV-2 Delta Variant Virus-like Particles, or in short VLPs, and its application in development of enzyme-linked Aptasorbent assay, or in short ELASA. So for information, I'm supervised by Dr. Siti Farah Alwani and Associate Professor Dr. Wan Siuk Moy from UITM, and also the Associate Professor Dr. Chia Swetlin from UPM and Associate Professor Dr. Chitarata Marimutu from USM. So we go straight to the introduction. Allow me to brief you a bit about this study. So when we talk about SARS-CoV-2, we cannot run away from the facts that this virus caused coronavirus disease 2019, which is in short COVID-19. So I believe most of us nowadays already aware about COVID-19, but let me brief you a bit that as of September 2025, approximately 700 million positive cases had been reported worldwide. And these numbers accounted for more than 7 million fatalities according to WHO 2035. And in Malaysia itself, over 5.3 million cases, contributing to over 37 deaths were reported. So for information, even though most of us already moving on from COVID-19, but trust me, each and every day, some people actually died from many parts of the world. So when we talk about SARS-CoV-2 and we talk about the virus, another fact that this virus actually is related to emergence of SARS-CoV-2 variants. So what is SARS-CoV-2 variant? It is actually the virus isolates or the virus itself that have one or more mutation as compared to the native or wild type. So wild type is actually the actual virus, the original strain that were isolated in Wuhan city in the end of 2019. And from there, the virus mutated, leading to the emergence of various SARS-CoV-2 variants. And for information, this is a bit of timeline of a major variants found or observed in Malaysia. And this mutation, we need to know the emergence of variants actually have some implications, especially on increased transmissibility, immune escape, as well as reduced vaccine efficacy. And not to forget about diagnostic challenges in which some kids during the emergence of this variant actually showing false negative. And in order to address this, WHO actually classified this variant according to their impacts toward the public health concern, classify the variant into certain classification into certain classes depending on its impact. And for this study, I'm focusing on Delta variant, which was isolated first in Malaysia in June 2031. And at that time, it was classified as variants of concern, which is considered to be one of the variants with the most devastating effects toward human beings. And this study, one of parts of this study involved the virus-like particles, which is shown, which is actually acknowledged in my title. So what is virus-like particles? VLPs in short refer to the particles which actually exhibit similar properties as the virus. In simpler words, basically the VLPs from the outside, it looks exactly similar like the active virus, like the actual virus, but it is non-infectious due to the absence of its genetic material, as shown in this figure right here. And for your information, VLPs have wide arrays of applications, including like drug carrier, gene therapy, vaccine development, and so on. And not only SARS-CoV-2, VLPs have also been extensively studied in many other viruses as well. And switching to the next focus of this study is actually aptamer. So what is aptamer? Aptamer are single-stranded nucleic acid, so it can either be DNA or RNA that can form tertiary structures which can actually bind towards its target molecule. So these properties or this action of the aptamers actually is a representative of the lock and key model of antibody towards its target. So due to this, aptamers can be referred or is usually referred as the synthetic antibody. However, despite it is known as synthetic, it actually have several advantages, being smaller in sizes, cost-effective, no batch-to-batch variation, and low to no immunogenicity as compared to the actual or monoclonal antibody. And in this study, like some of you might be wondering, what is ELASA or enzyme-linked Aptasorbent assay? So just like the name suggests, it might trigger you something. I believe most of you are quite well versed with ELISA, in which ELISA we are using antibody as the detection or capturing agent. In this case, we are switching or we are shifting the focus towards aptamer to serve the same purpose as the antibody, or in other words, it serves as antibody, it substitute antibody for this study. And these are some of the problem statements for the study. For your information, SARS-CoV-2 or the virus causing COVID-19, actually it requires a biosafety level 3 facility, which is for SARS-CoV-2 for its viral propagation. So this is required for us to conduct extensive study when it comes to this virus. However, for your information, in Malaysia, at least only 4 BSL-3 level 3s or BSL-3 facilities that are available in Malaysia. So this actually limits the required research that we need to do when it comes to this virus. And the next one, those impacts when it comes to SARS-CoV-2 variant emergence, like what I mentioned earlier. So that's why we focus on Delta variant, because by studying VLPs, by actually producing VLPs, we can actually study the immunogenicity or maybe the viral pathogenesis of this virus without the need of BSL-3 level 3. And due to the non-infectious of this VLPs, it is a safer alternative for us to work with. And the next one is for information, when for those who were infected with COVID-19, there were some treatments available for them. However, this actually monoclonal non-infectious, so it can be applied as a powerful tool to study the SARS-CoV-2 variants without the need of us applying or implementing the actual virus. So this is safer for us to work with, as well as it can also be used as a potential vaccine candidate. And with the non-infectiousness of these VLPs, we can now conduct or perform any related research when it comes to these variants to many facilities, including the one that we have here in Immb, without the need for BSL-3 facilities. So actually they eliminate the requirement of those high maintenance facility. And then when it comes to Aptamer in this study, we focus on in silico refinement, which is a computational based approach, a computer-based approach in which it offers a straightforward, it offers a cheaper method for us to define, for us to predict or refine or to improve the ability or the capacity of these SARS-CoV-2 aptamers. And the last one, for your information, in this study, we employ these aptamers to develop the ELASA, which actually offers a straightforward and cost-effective development of a highly specific and sensitive diagnostic tool for SARS-CoV-2. So these are some of my hypotheses, which again, I will actually go through this later. And we go straight to the research methodology. Okay, so basically this is a simple, I believe this is just a simple study design in which although we have 4 objectives in total, but we divided our lab work, we divided this project into 5 phases altogether. The first phase involved the construction of SARS-CoV-2 Delta variant DNA, in which we constructed the recombinant DNA carrying all those recombinant genes, all those required structural genes that are needed to actually produce our virus-like particles. So this involved the retrieval of all Delta variant sequences. We then aligned all these sequences and we generate or we constructed the recombinant DNA. Following that, once we already have the recombinant DNA, we proceed to phase 2, which is the production of SARS-CoV-2 VLPs using mammalian expression system. So with the availability of this recombinant DNA, we then transform the E. coli to actually to clone it, to propagate the plasmid, all right? And then we use it to transfect, or we introduce this recombinant DNA into Vero76 cell line, which serve as the mammalian expression system to produce the VLPs in this study. And following the transfection, we subjected the transfected cell to neomycin selection to only select the transfected cell or transfecting cells, and then we culture and we prepare the cell lysate from this transfected cell. And phase 3, in order for us to assess the antigenicity. So for your information, antigenicity, we can assume it as the similarity of the VLPs with the native virus. So in this case, we retrieve, in order to assess this, we retrieve convalescent sera from the recovered COVID-19 patient. Before this, they were infected with COVID-19 patient, and once they recovered, we collected their sera. We screened for the presence of anti-SARS-CoV-2 antibodies, and we used the antibodies that already known to actually detect the presence, can actually recognize the actual virus and see whether this antibody can actually detect or determine or maybe recognize our VLPs. And phase 4, which I assume one of the biggest part of this study, involved the refinement of aptamer, in which we retrieve anti-SARS-CoV-2 aptamers or aptamers targeting the viruses, and we subjected these aptamers to aptamer refinement. In total, we have 4, which we are going to go through one by one later. And following this refinement, we then subject this aptamer to ELASA, which is, as you can see here, we actually developed 2, direct and sandwich ELASA. And the final phase is actually a much simpler one, in which once we already know that the aptamers can detect or recognize our VLPs, we then use this aptamer to purify our VLPs from the sample, in which this is a novel method. We actually integrate the the capture assay, which is using magnetic beads, but this time around using aptamer. And moving on to results and discussion. So for the first phase, which is if you remember previously, I mentioned about the retrieval of all SARS-CoV-2 Delta variant sequences. So in total, there were 6,300 something sequences isolated in Malaysia from 1st of August to 31st December 2021. And once when we aligned these sequences, you could see actually for Delta variant, there were 15 highlighted mutations by WHO. So all these mutation actually being a characteristic or a criteria for Delta variant. So when we aligned these sequences, we then check for the highest prevalent or maybe the mutation with the highest prevalence when it comes to these isolated sequences. And out of 15, we could see at least 10 of them actually being found or was observed in most of the sequences, at least half of the isolated sequences. So for your information, T19R, how to read it, is actually the mutation is originally T, amino acid T, at position 19 of the spike protein, and then following the mutation, it was mutated to R, which is T, but now it's becoming R. So some of this mutation, it contributed to some of the viral fitness of the virus. One of them is T478K, which is this one, T478K. So this is one of the most prominent, one of the most prominent mutation when it comes to Delta variant. It allows this virus to actually have the increased transmissibility, and that's why we can see some of these mutations are observed in higher prevalence as compared to the other, because this virus, when they mutate, they tend to actually sustain or maintain the mutations that allow their viral fitness, and they tend to actually remove or eliminate the mutations that are not actually contributing to their viral fitness, as simple as that. And then when we constructed the recombinant DNA, we are not only focusing on all those spike mutation. We are not only focusing on spike mutation. As you can see here, we also included membrane and also envelope protein. So these two proteins, they are required as a minimal requirement for us to construct the VLPs. So the action of the membrane protein as well as the envelope protein, that actually mediates the interaction which allows this virus or which allows this VLP to be transported out of the cells, which actually in some ways it replicates or it actually represent the actual virus pathogenesis. And then we also include this one, EGFP or enhanced green fluorescent protein, which serves as an indicator, which serves a visual marker for us. For example, basically this is a DNA map, and this is where EGFP is located. So this is what we use to transfect our cell, And EGFP allows us to observe whether this protein have been produced or not, because EGFP, if it is being produced, it will actually allows the cell to fluoresce under fluorescent microscopy. And this is the observation. For those who are familiar with cell culture, you might think that this one actually have been dyed or stained. But trust me, this is not a stained cell. This is just a normal cell following transfection. And all these fluorescing cells were actually due to the expression of the EGFP gene like I mentioned earlier. So this one, we transfect the cell and then we just subject it to fluorescent microscopy with any stain, and we can observe like this one. And figure 4.4 shows the increased fluorescent intensity of the transfected cell. And from here, we deduce that if the fluorescent protein is being expressed, so we deduce that the upstream protein, which is all this membrane, this is the membrane envelope, and as well as spike protein, which are needed for the VLPs production, are also being expressed. And interestingly, in this study, we found this observation, or we observed this one, which is known as synthesia, is actually a hallmark CPE, or hallmark observation for SARS-CoV-2 infection. This kind of cells, or this kind of observation, is usually found or usually observed in pneumocytes, or basically the lung cells, all right, when some people or when individuals got infected with COVID-19. And this case, because we are only transfecting the cells, we are only producing the VLPs which are non-infectious. However, these non-infectious VLPs actually sustain or maintain the pathogenesis of the CPE of the actual virus. So this is interesting for us, and this figure here shows the an observation which what we deduced was to be a vocalization, which is also formed through the actions of these VLPs. And a bit for me to add on this synthesia. So basically, synthesia is actually a large multinucleated cell, look like this one. So this is actually being formed through the action of the spike protein of the viruses, in this case the VLPs, which actually mediates the interaction between neighboring cells. So all those neighboring cells, they will they bind or they join together to form a large multinucleated cell like this one. All right. And then in order for us to select only the transfected cell, why do we need to select? Because we only want the cell that are actively producing or expressing VLPs to be cultured. We subject them to neomycin selection. So in order to determine the actual concentration of neomycin to be used, we perform neomycin kill curve assay using a normal cell. So basically, a different concentration of neomycin were used to treat the non-transfected cell or the normal cells. And then following day 21 or 21 days after treatment, we could see as low as 1.1 milligram per milliliter neomycin was able to actually kill most or all of the cells. So that's why we choose this concentration to select our transfected cell. And these are the observation. Following the neomycin selection, basically following transfection, we then subjected the transfected cell to neomycin selection using 1.1 milligram per mil, just like what we determined before. And then from here, you could see following day 1 or 24 hours after treatment, up to day 12, up to day 9, basically the cells or transfected cells undergo mass or massive cell death. So this is actually due to elimination of most of the cells. So it's either the cells are not transfected, or it's also some of the cell may not be able to actively secrete or does not actually actively take up the plasmid that we transfected earlier. All of these cells will be killed during this process, and from day 12 onwards, only the surviving cells, which we assume which should be the cells that only have the plasmid actively expressing the virus-like particles, will actually survive, and from there they will propagate. And we move on to phase 3, which is now we already have our VLPs. So what's next is like we need to assess whether these VLPs, the one that we produce, is similar to the actual virus, because that's the main aim. That's one of the aim of us actually producing the VLPs. So in order for us to assess, first we screen the convalescent sera. We retrieve some convalescent sera from recovered COVID-19 patient, and we screen this convalescent sera for the presence of NTA spike IgG, or basically the antibody against spike protein of the virus. And as shown here, actually all of the sera samples, all of the sera showed a positive showed seropositivity or positive results for the presence of the anti-spike IgG. So this allows us to use this sera for determination, because you need to know once once these sera actually have the antibodies that can detect the actual virus. So this sera sample can allow us to determine whether the VLPs that we produced earlier actually represent or resemble the actual virus or not. And that's when we proceed to this one, which is the antigenicity assessment of the transfatted cell lysate and supernatant by dot blot assay. So for your information, dot blot assay more or less it serves the same principle like a Western blot, just that Western blot we need to do the SDS page, we need to linearize, and then the protein will be fragmented according, or will be, sorry, the protein will be fermented according to their sizes. But this time around, .assay, we just dot the VLPs on a PVDF membrane, and then we add the antibody that we got from the sera, and we add the anti-IgG to actually have the HRP reaction later. And dot here, or the intense dark spots here, showed actually a positive results, which determined or which show that our VLPs actually can be detected by this sera, by the antibody that can be found in the sera, which in return tell us that, which in return we deduce that these VLPs are actually of high resemblance of the actual virus. And next, in order to further confirm that, we proceed with indirect immunoperoxidase, which is another study using the sera sample, using the sera which contain the antibody, in which in this case we were using lab samples, and then we treat them or we incubate them with the antibody from the sera sample, and the antibody will actually bind to all these parts of the cells that actually have the VLPs. And following the colorimetric detection, which addition of the colorimetric reagent, we could observe these reddish brown colored particles, which actually determines the positive results or positive observation for indirect immunoperoxidase. And here we could see that not only this cell actually actively expressing the VLPs, but we could also know the localization of these VLPs as well. And then we move on to one of the biggest phases on this study, which is phase 4, refinement of aptamers. So the first one, we proceed with the retrieval of aptamers. So in order for us to determine which one actually have the aptamers, we subject it to two inclusion criteria, which is in silico and in vitro. So for your information, aptamers can be developed through in silico, which is more or less through structural modeling, and in vitro selects, which is it involve PCR, involve the amplification and so on in vitro. So in this case, we choose or we retrieve the aptamers which are being produced from both of these in silico and in vitro. And then in total, we have actually 5. 3 from in silico, 1 from in vitro, which we're targeting RBD, which is one part of the virus, and another one is SARS-CoV-2, which we're targeting pseudovirus. Pseudovirus is actually not an actual virus, more or less the idea is more or less like a VLPs, but the production is a bit different. It's like a different virus, but actually they are presenting SARS-CoV-2 antigens or protein. And then the first one, we proceed to, we subjected these aptamers to rational truncation. So allow me to brief you a bit. This is basically the secondary structure of aptamer. This is basically a normal one, and in which there are two main parts of the aptamer. The one that I highlighted here, the one in the red box, is actually known as the loop, and the one that you can find here is the stem or the duplex region. So when it comes to aptamer, the loop region is always known, basically it is known to be the hotspot of the binding, or when it comes to antibody, it serves more or less like epitope, or the region, the site where the aptamer binds towards the target. So truncation was done only on duplex region because we do not want to disrupt this region. And then following the removal of nucleotides from the duplex region, we subject all these aptamers to secondary structure prediction. The idea of rational truncation is to remove the duplex region to make it shorter, but not to actually disrupt the loop region. So that's the idea. As you can see here, the first aptamer, aptamer 25, can be truncated up to 11 mer or 11 nucleotide from its actual size or actual length of 25 nucleotide. So this actually have some, of course, some advantages. One of them is it is becoming more stable. Second one, of course, the cost will be lower because we are synthesizing some aptamers that are shorter in length. And we did the same process for the other aptamers, which is aptamer 27, 31R, and AR10, which I'm going to actually summarize later. All right. And then the next refinement, we did the base substitution. So like I mentioned earlier, this is actually the duplex region of the aptamer. So following truncation, some of these aptamer actually have a lower delta G energy or delta G value. So this delta G value actually indicate the structural stability. So to improve its structural stability, we change or we substitute the base on the duplex region from ET to GC. As you can see here, the delta G from 2.16, it was reduced to 0.54. Again, the lower the delta G energy, basically the higher the structural stability. So that's why we did it for these aptamers. And interestingly, if you remember before, this is actually one of the aptamer, which is aptamer 25, that we could only truncate it up to 11 mer or 11 nucleotide in length because it couldn't retain its loop structure if we truncate it any further. But with the base substitution, we can actually introduce or we can actually truncate it more, so allowing us to have a shorter aptamer before it was base substituted. So the same was observed with aptamer 31. This is without the base substitution, and this is with base substitution, it retained its loop structure. And the last one, actually, we proceed with 3' or 3' polyadenylation. Basically, what we did, we added the polyethyl here at the 3' end of all this aptamer. So more or less, this serve as a structural stability to actually increase their resistance towards degrading enzymes in several conditions, and as well as this will allow us to implement these aptamers for subsequent ELASA. All right. And this is actually the summary of what we did. So basically, the first one, like I mentioned earlier, truncation, even though we managed to shorten the length, it actually elevates the delta G values. So basically, the increase in delta G values, as you can see here, aptamer 25, originally it have -8, but following truncation, it was increased or it was elevated to 1.17. So it was becoming more positive. So basically, higher delta G value means it is less stable. And however, only one aptamer, which is aptamer R here, following the truncation, the delta G energy or the structural stability was not compromised. So from 40 mer or 40 nucleotide, it was actually truncated to 29 mer with the removal of 11 nucleotide, and it actually maintained its structural stability. So that's why we proceed with all these refinements. As you can see here, following the truncation only, the delta G will be increased. So which means the structural stability is lower. But following the base substitution from 2.61, it was reduced to 0.54. So after base substitution, this aptamer 37T, actually becoming more stable, and following 3' polyadenylation, it was becoming more negative. So meaning that it was becoming more stable. And next, we tested all these aptamer for direct ELASA. So this is basically direct ELASA against the recombinant spike protein. As you can see here, all of these aptamer, regardless of some of them maybe have a lower OD reading, but most of them can actually recognize the spike protein using direct ELASA. And from here, we choose two of the best aptamer, which is aptamer 25 and aptamer AR10, actually, which actually have the highest OD and of course the highest significance, right, for us to proceed with the subsequent sandwich ELASA, because in total here we have 10 aptamers, so we cannot proceed with 10 due to limitation of cost and time. So we choose only two of the best aptamer for subsequent sandwich ELASA. Okay, so this is where, as you can see here, not only that we determined the the t-test, the significance through unpaired t-test, we also determine the signal to noise ratio, because some aptamers they might have a higher OD reading, but when we compare with the background reading, actually they have a diminished signal to noise ratio. That's why we choose two of the best, which is aptamer 25. This is the signal to noise ratio, and and AR10, aptamer RT with 8.625, which is two of the best aptamer with the highest signal to noise ratio for subsequent. And then these two aptamers were then subjected to another refinement, which is multimerization. So basically, multimerization is like we combined two aptamer into one using a poly-T linker in between these two, as you can see here. So A here is actually made up from two aptamer 25, and this one here is made up from two aptamer RT. And the C here is actually a combination of both aptamer 25 and aptamer RT. And as you can see, again, multimerization further decrease the delta G energy or delta G value, which indicate the multimerization further improve its stability. Next, we subject these aptamers, we subject these capturing aptamers to sandwich ELASA, in which we use detection aptamer, two type of detection aptamer, which is 25 and RT. So we choose these two because these two actually showed the highest OD reading and the highest signal to noise ratio during direct ELASA. And when we use all these sequences or all these combinations, or in total there were 14 combinations, as you can see here, different capture aptamers against a single detection aptamer 25, and here 7 capture aptamers against a single detection aptamer RT. So from here we know that only one actually have the best signal to noise ratio, which is this one, okay, which is in which we use capture aptamer homomery RT, which is consists of two aptamer RT combined together, multimerized together, and then we use aptamer 25 as its detection aptamer. So these are basically some of the discussion about that part. We know that multimerization from here, it actually increased the stability, and of course it also increased the specificity or detection or affinity of the aptamer towards the target. In this case, it is the recombinant spike protein. Okay, but interestingly, if you can see here, there are actually two aptamers that are being multimerized together. One is homomeric RT, which is RT and RT we combine together. Another one is homomeric 25. One is 25 and another one is 25. We combine it together. Only one, which is homomeric RT, managed to have a higher signal to noise ratio. Meanwhile, homomeric 25 do not have any significant difference between the detection and its control. So what can we deduce from here? Basically, the combination of homomeric aptamer 25 was due to the combination of non-truncated aptamer. So basically, it's a long aptamer. Then when we truncate it together, this may cause some strict hindrance or some some unnecessary sequences that may actually results in the low binding or low affinity of this aptamer towards the target. However, for homomeric RT, which is consists of two truncated aptamer, so we already removed the unnecessary duplex region, unnecessary region that may interfere with the binding of the spike protein. That's why it managed to actually show demonstrated a high signal to noise ratio. All right, and then this one is actually sandwich ELASA. Lastly, we use this sandwich ELASA. We determine the best combination, which is the homomeric RT as the capturing and aptamer 25 as the detection, and we subjected this combination or this pairing for the detection of VLPs from different cell lysate, from different combination. So once we have the transfected cell lysate, we treated this cell lysate with different conditions. First one, we're using RIPA. Second one, we're using Triton X. Next, we're using freeze-thaw. So for information, freeze-thaw is a common practice for us to actually isolate the viruses or to harvest the virus. And the last one is actually the supernatant. We filtered the supernatant from the transfected cell. We subject all of them to the detection of using sandwich ELASA to determine which one actually give us the best or reading, which in return shows the best harvesting of the VLPs. And from here you can see VLPs RIPA or the transfected cell lysate treated using RIPA managed to give the highest OD reading and as well as the signal to noise ratio. So from here we deduce that the treatment of RIPA was the best one to actually harvest the VLPs and intact VLPs, and actually it maintained its uh, structural correctness. So it could be actually detected by our sandwich ELASA. And to further determine whether our sandwich ELASA, the one that we developed, actually have a good diagnostic performance as compared to the commercial kit, we actually compare this. On the right side is actually the sandwich ELASA, the one that we developed, and on the left side is sandwich ELISA, which is a commercial one, a commercial ELISA kit, the one that using antibody, and the right one is using our aptamer. As you can see, even though sandwich ELASA managed to give us a higher OD reading, however, when we compare signal to noise ratio, our sandwich ELASA managed to give us a higher signal to noise ratio, and it is more significant between the VLPs and of course the background reading. So from here, what we can deduce is that even though our sandwich ELASA may not be that high, may not be sensitive in terms of its OD reading, but it can actually distinguish between two samples, which actually one have the target, in this case VLPs, and the other one without the target, which is the control cell lysate. So this further support our deduction that this kit actually have a high diagnostic performance, and it should be translated to future diagnostic kit later lah. And then the last one, in order for us to determine whether this kit actually have a higher or low sensitivity, so we subject them to limit of detection and limit of quantification. So these are actually the graph, and following calculation, what we managed to get is limit of detection is 40 nanomolar, which is as low as 40 nanomolar of the protein of the SARS-CoV-2 could be detected by our kit, and LOQ, which is limit of quantification is 42 nanomolar, in which we need at least 42 nanomolar of protein for it to be quantified using our kit. All right. And then for your information, this value is not it does not shows a really low sensitivity, which is, for some people it may not be really how do I put it, it might not be really sensitive. However, this kit, we have this kind of LOD and LOQ. It is actually uh, advantages. One of its advantages, following times the virus tend to be actually replicate in their body. So this is actually we can know, for example, if someone is being affected by COVID-19, and if we could actually use this kit to quantify or detect the virus, so we know that these people are typically associated with moderate to high viral loads, and these often or commonly translate to severe clinical manifestations, which it also helps us for a proper management of the disease. And lastly, which is the last phase, okay, so last phase is development of aptamer-based capture assay. Once we already know that this aptamer is specific against the VLP, specific against the virus, we then implement it for aptamer-based capture assay, just like what it did here. So it can actually capture our viruses, and we use magnetic beads to actually purify the viruses. And this is the SDS page that show intense band roughly at 300 kilodalton size, which actually shows which we deduce to be the VLPs of interest lah. And then, however, there is another one that is here, which is roughly around 100 kilodalton and intense band here. So this is actually what we deduced it was S2, which is the spike protein was cleaved. So for information, in this case, we use the Delta spike protein, and Delta spike protein, one of the mutations is to allow this spike to be cleaved for a more efficient by cell entry. So that's why we managed to see this one is what we deduce to be the whole VLPs, and this one should be the cleave or the fragmented spike protein. right? And as for conclusion, implementation of mammalian expression system successfully produced SARS-CoV-2 Delta variant virus-like particles as confirmed by several immunoassays, which we were using the sera sample which contain the antibody targeting the actual virus. And next, the use of anti-SARS-CoV-2 antibodies. This is what I mentioned earlier. For convalescent sera, verify the structural integrity because we already produce VLPs, and these VLPs can now be detected by the antibody that can detect the actual virus. So that actually verified the structural integrity of the VLP. And the refinement of the aptamer enhance the binding through in silico strategies in which we use computational method to truncate, to base substitute, and to add the polyaethyl for these aptamers. And this actually has been proven that it can actually enhance the binding of these aptamers towards the virus, towards the target. And the last one, aptamers multimerization, just like what we did for our sandwich ELASA, can increase binding multivalency because it have more than one sites, more than one capturing sites for it to capture the target in this case, and improving the specificity of the target capture. And these are some limitation and recommendation of this study. This one of the limitation of this study is not is that it do not involve any wild type, any actual viruses, because we focus on the on the virus-like particles, and we already assessed the antigenicity of these virus-like particles. And the next one is in vitro validation, because we were working with lots of aptamers and we only managed to test on some of them. So for information, we tested at least 14 different concentration, 14 different concentration of aptamer. And in total, actually, it is more than that lah. Up to 50 different concentrations can be tested. But we rely on predictive modeling, which is the in silico punya approaches. And from here, there is a possibility that some aptamers that have a better in vitro performance might be omitted throughout the process. And here are some recommendations in order for the VLPs to be used as a potential vaccine candidate in future, or maybe a targeted drug carrier. Comprehensive evaluation and immunogenicity as well as biosafety should be done on the VLPs to use it for diagnostic and beyond. And the next one, because this study focusing only on Delta variant, so multiple variants of SARS-CoV-2 can be used or can be targeted to enhance the diagnostic resilience and relevance of this kit. And the last one, basically this is the idea of the kit that we developed. The additional investigation into its thermostability, shelf life, and storage requirement of the developed ELASA is needed for us, for it to be translated into, I would say, into a good diagnostic kit, or into maybe a point of care testing as well. And these are some of the publications throughout my study, and more in draft, inshallah. And these are some of the achievement and recognitions that we managed to achieve throughout this study. And lastly, I would like to acknowledge all my supervisors, Dr. Steve Arawani, Associate Professor Dr. Wang Sik Moy, Dr. Chas Welin, and also Dr. Sidatan, as well as Immb, AMDI, my group, DMM, Laboratory, as well as Medipa. So that will be it from me. Thank you very much. Yeah. So
Okay, thank you very much, Mr. Amir, for the well done and insightful presentation. So I would like to open the Q&A session. Usually it's the one asking. So today he will be answering. So please ask him questions.
yeah, please, feel free. Yeah, yes, Dr. Afiq.
Amir Muhaimin, thank you for the presentation.
Okay, welcome.
Now, can you go back to the method that you chose for this project?
Okay.
And can you tell me
Which one?
what the method?
Wait,
The method design and do.
this one?
This is your study design.
Yeah.
Okay. Why do you choose this particular study design or this methodology instead of any other alternative? Is there any similar to?
Okay, so thank you, Dr. Afiq, for that question. So basically for this study design, this is a general one. We have 5 phases altogether. And in order for us to develop a virus-like particles, I'm afraid that for the first phase and second phase, this is basically the idea of what we should do lah. Right? We need to actually design the recombinant DNA, and we need to produce the VLPs before we determine the antigenicity. But for phase 4 and phase 5, for this study, we were focusing more on aptamers, which is synthetic antibodies. Why? Because we want to shift the focus from monoclonal antibodies to aptamer. So, based on your question, maybe an alternative way of doing it. So maybe, for example, if we are going to produce an ELASA platform, a diagnostic platform, another way to produce it, to use it is maybe using hybridoma, which is we produce monoclonal antibody rather than aptamer. The first one. The second one, because this study we use ELASA as the platform, and another alternative for ELASA, it will be biosensor, LFA, which is lateral flow assay, and some other diagnostic platform as well lah. One of the reasons why we choose ELISA is because it is most straightforward, the cheapest, and of course it is one of the non-laborious lah.
What about the decisiveness of the ELISA itself?
Okay, so for this study, we actually we tested, if I may share, we tested the LOD and LOQ. So I will just show some of the main results lah. Wait, Dr. Afiq. Okay, so for this one, we compare the ELASA that we developed with the commercial ELISA. On the left side is actually a sandwich ELISA that we commercialized. It was from ELAB Science that I believe most of us prefer ELAB Science like. Okay, so this is ELAB Science protein S1-RBD ELISA kit, and this is our sandwich ELISA. So in terms of diagnostic performance, in terms of the specificity, we could say that even though our OD reading is lower as compared to the sandwich ELISA, but our signal to noise ratio is higher. So this is actually really important for us to distinguish between the target is there or not, especially in a more complex matrices, for example, like the sera and so on, with all those interfering proteins. This is one, and this is the second one, which we determine the limit of detection and limit of quantification. Like I mentioned earlier, even though our limit of detection is not as low as it should be, but then basically this is actually advantageous when we want to actually determine or we want to actually classify the severity of the disease, because people with severe disease usually have a higher viral load as compared to people with a mild disease lah. So this is those two actually, one determine its specificity in which we can detect the virus, just like the actual kit, commercial kit, and this one actually determine its sensitivity lah. Yeah.
So based on your result, it is is it enough to be translated into the clinical setting?
Yeah. Okay. So very good question. So that's what we are working. We plan to work on later, because we already have the aptamer, and in this study we already know that this aptamer is highly specific against the target, which is the virus. So what we are going to do next, because ELASA is only the starting point, I would say, is only the beginning of the diagnostic platform. So in order for it to be translated into clinical settings, especially when we talk about point of care testing, those that can be done by ourselves. So that one is usually like RT care that we use for COVID-19 kan. So that one need further, I would say, further optimization. How can we use this aptamer to that different platform? But actually we are looking forward to that lah. Apart from LFA, the one like RTK, we are also looking forward to actually to implement it, to translate it into biosensor as well. So biosensor will work more or less like a glucometer, in which we just put our samples and we read using a device lah. Yeah. Yeah. If there is phase 6 lah, doctor. Yeah. Thank you. Susah je.
Thank you, Mr. Afiq.
Doktor.
Sorry, guys. Other questions? Salina? May I know what you will be using the fluorescent intensity?
Okay, so fluorescent intensity, actually we capture the image using fluorescent microscope, then we subject it to image J lah. Image J. So I forgot to actually mention that, but we determine the fluorescent intensity using image J. Yeah. Okay.
Thank you, Salina.
Cepat, macam banyak soalan ni.
I have one question.
Okay,
So,
one je.
well done for the presentation,
Okay,
Muhaimin.
thank you.
So, since we know COVID-19 nowadays is under control, right? So, how do you see the relevance of this research to the current situation? Okay. No, I have.
Okay. Just now you said only one, but okay, two. Okay.
Yes. Okay. Do you think this method can be applied to detect any virus like influenza right now?
Okay, so thank you, Roha. So the very first question is actually one of the most famous question that I got or my team got when we presented innovation conference anywhere. Is it still relevant when it comes to COVID-19? So it is either yes and no, I would say, to be honest, but I will answer yes first. So yes first, I'm not sure if you all ever come across this paper. So if you look through, there are many papers that they are trying to link COVID-19 with mental or with a neural disorder. So some of them actually, some scientists, they actually link COVID-19 along COVID-19 because now I would say COVID-19 around more or less 5 to 6 years following its first detection. So some of us might already recovered maybe 5 years from COVID-19. But some people, they say that they are predicting using the machine learning and so on. They are predicting maybe 10 years from now, people who recover from COVID-19 will start to have a higher possibility of Alzheimer's, dementia, and so on. So it's one of that lah, because we need to know how people are getting affected, and sometimes the higher viral load, or for example, if people are not getting treated, this actually increase the risk of the what I mentioned earlier. The second one, even though in Malaysia we assume that it is not really relevant, but in the past 7 days, at least 1,000 of people are still dying from COVID-19 worldwide. Yeah. So, and for diagnostic wise, in which for now we are only focused on diagnostic, detect whether people have it or not. But the next aim is for us to broaden the horizon to maybe to therapeutics, because what is the most important now is for us to treat the people rather than diagnose lah. So that's will be my answer for the first one. And the second one, when you said about for the other viruses, yep, when we want to produce VLPs, for example, we can use it for other viruses. Okay, but for ELASA, that one is have a broader application in which we can use it to detect anything that come to your mind. It can either be hormones, it can either be chemicals, it can either be viruses, bacteria, and so on. As long as we know how to refine or determine the specificity of this aptamer towards the target lah. Yeah. Okay. Dah.
Thank you, Roha. No other questions?
Semua.
Semua dah keluar dah.
Aduh, semua tak ada. Beri lah.
Dr. Wang, you have any?
Doktor. Later lah, Doktor. Okay. Dr. Wang would like you to grill me. That's what. Kan, Doktor kan? Kembali later. Okay, sorry, Doktor.
Okay, so if that's the case, I would like to conclude.
Online?
No questions from the online participants.
No.
One comment from Fatihah Roni, very good presentation and all the best for your Viva. Okay, so if that's the case, I would like to conclude our session today. Congrats and thank you very much to our presenter, Mr. Amir Muhaimin. Okay, for the online participants, you may click on the link I've provided in the chat box for the attendance form. Thank you very much. I will share the recording on our YouTube channel soon.